Okay, so the next chapter is about DNA and protein synthesis. And this is one of the very important chapters in this unit. This one and the next one, which is a reproduction. So you will see, because it's in the core of this unit, you will see a lot questions in those two units specifically. This one, DNA and protein synthesis, and the cell reproduction, which would be the next. I would say it's yeah, it's going to be next week. But for now, let's do the DNA and protein synthesis, and this is very important. So pay attention. We are going to talk about the structure of the DNA. What does it mean first? DNA stands for deoxyribo. This is one, D, deoxyribo. N, nucleic acid. What's deoxyribo? Ribo is ribose. This is what it means, a sugar called ribose. And this sugar is in the RNA. So in RNA, we will talk about it. RNA is ribonucleic acids, which means ribose, nucleic acid contain ribose. How about this one? Just put deoxy. So did you get this part? RNA is ribonucleic acid. What does it mean? It's a nucleic acid that contains sugar called ribose. This is R, ribose, ribo. Now, what's D? D is the same, but instead of ribose, it's deoxyribose. And what's deoxy? D means without. So it's the same thing, you just removed oxygen. That's it. Ribonucleic nucleic acid contain ribose. If you take this ribose and take oxygen out of it, it will be deoxyribose. Okay, I hope this part is clear. So that's where the name came from. And that's why ribonucleic acid, RNA, all the types are called ribonucleic acid, nucleic acid containing ribose. Deoxyribonucleic acid is also nucleic acid, but it has deoxyribose, like this. It's a nucleic acid contain deoxyribose instead of ribose like this. And we will talk about the different types of RNA, which is ribonucleic acid. There is messenger, ribosomal, and transfer. And then we will talk about the protein synthesis in different levels. And we will also discuss a little bit about the DNA replication, which will be covered more in the next chapter. So what's a DNA? We talked about this before when we did the cell, the structure of the cell, and we should remember that DNA is the genetic material located within the nucleus, and it contains the inherited traits. Everything that determined the inheritance, what did you get from your parents, was written here in the DNA. This is one, inherited traits. That's number one. Number two, it is responsible for protein synthesis. So it contains like the blueprint for all proteins that we use inside our body or that we need inside our body. The manuscript, the blueprint, okay? And I always say it like this, like the nucleus is the manager's office. And when you are in the office, you have like a storage or a safe, and you put your important stuff in there, like original instructions of how to do something. If this is a factory, let's say TV factory. So the original script, the blueprint of how to do TVs, you cannot give it to anybody. You have to keep it in there. You keep it in the safe, you keep it in the office so nobody can take it. Then, if that's a factory, how can the workers work on the units, different units, and make TVs? Yes, I will give them a copy of the part that I want them to not to make specifically. And this copy will be messenger RNA, the first one. So I have this whole blueprint of all types of proteins. I will never allow anybody to take it. It's not going anywhere. All what I'm going to do is I want you to make the knobs of the TV, let's say. I'm going to give you a copy of this part only that specify how to make the knobs. Okay, and if you apply that to the proteins, I have this DNA contain all instructions for all proteins. I want to make protein number one, whatever it is. So I'm going to make a copy of the instructions of how to make protein number one only. And this copy is called messenger RNA. So the instructions, every group of instructions is called a gene. So what's a gene? A group of instructions within the DNA. It's important to know that DNA is not just scattered within the nucleus. No, it's arranged mainly in the form of chromosome. What's a chromosome? It's the DNA that is double-stranded and it's arranged in a specific way to be stored in the nucleus. And it's always associated with a protein. If the cell is going to divide, that same chromosome, the same one, is going to change its structure and become more visible and thicker, and we now call it chromatin. So chromosome and chromatin are basically the same thing, but the only difference is chromosome is where the DNA arranged all the time. The exception to that, if the cells are divided, the chromosome change and become chromatin. It's change, it's it's just configuration, not the structure. So and it become its DNA associate with a protein. So if you look at this is like the DNA. It's arranged in the form of what? Chromosomes. What's a chromosome? It's not just a double-stranded DNA molecule. It's with protein as well. With I don't know why they don't have it, but this is what it is. Both of them have protein. So chromosome and chromatin, both of them are DNA with chromosome with protein. Yes. What's the difference? The configuration. Chromosome is more tightly coiled, and this is where the cells are not replicating. The cells are not duplicating. It's not the cell division yet. This is the main life of the cell in the form of chromosome. When it is the time to divide, when it's the time to duplicate and divide, now this tightly coiled DNA with protein that we call it chromosomes become a little bit looser uh, or start to change its configuration and become thicker and shorter, and now we call it chromatin. So the key word here is cell division or no cell division. During cell life, it's chromosome. If it is a time for cell division, it become chromatin. Follow with me as we go because we will go deeper and deeper. So follow with me. So so far we're talking about inside the nucleus we have DNA. DNA is a blueprint for the proteins and inheritance. DNA is not just scattered. No, it's arranged in the form of chromosomes. If the cells are replicating, the chromosomes become chromatin. On the DNA, there are segments of DNA. These segments are called gene, like this. This is a gene on the DNA. This is a gene. There are a lot. You see yellow, red, green. All of these are different genes. And if you magnify a little bit, you can see it here. Like from here to here, this is a gene. Okay? The gene is the instructions to make uh, certain, and it contains certain structures. We have a lot of those inside the DNA. So do not confuse genes with DNA, with chromosomes, with chromatin. DNA is a double strand. It contains a lot of genes. DNA is arranged in the form of chromosomes, and the chromosome change and become chromatin at the time of cell division. Now, the DNA, the structure of this nucleic acid, it's made of the building blocks of the nucleic acid, not just DNA. The building block of the nucleic acid in general is nucleotides. Do you guys still remember? I hope you still remember when we talked about carbohydrates. Carbohydrates is polysaccharide. The building unit is monosaccharide like glucose. Fat or lipids, this is the polysaccharide. Its building unit is fatty acids. Protein is a polypeptide. The building unit is amino acid. Here, nucleic acid is the polypeptide or the big form. The building unit is nucleotides, and you need to know that. Nucleotides. What's a nucleotide? Nucleotide consists of, and we're going deeper, so follow. DNA consists of a nucleic acid that consists of multiple nucleotides. What's a nucleotide? The nucleotide is nitrogenous base, one nitrogenous base, plus phosphate, plus sugar. And the sugar here is called deoxyribose. How about RNA? It's just ribose. And this is one of the main differences between DNA and RNA. How about the RNA? You will see it's coming. It's also a nucleic acid, so it's also made of nucleotide. It's also the nucleotide is nitrogenous base, phosphate, and sugar. So both of them, DNA and RNA, both of them are nitrogenous base, phosphate, sugar. Then what's the difference? The sugar is different. The sugar in the DNA is deoxyribose. The sugar in the RNA is ribose, which is almost the same, just removed oxygen out of the ribose. The other thing is nitrogenous base. There are 4 types of those: adenine, guanine, thymine, and cytosine, AGTC. There are 4 types. This one, when we do the RNA contains 3 out of those 4, but thymine, they replace it with something else called uracil. All right. The other thing is these nitrogenous bases, ATCG, these nitrogenous bases, remember the DNA, the D should remind you with double strand. D means deoxyribose, but it should remind you, just a reminder, that it's double. DNA is double, DD, remember. So it's 2 strands. So how the 2 strands stay with each other? 2 things. Number 1, these nitrogenous bases, they fit together. So here is one strand, and here is the other strand of the DNA. Both of them are nucleic acid. Both of them contain nitrogenous base, phosphate, and deoxyribose. Yes. How the 2 strands stay with each other? It's a double strand. They stay together by fitting each 2 correspondent to each other of these nitrogenous bases. So let's say this is strand number 1, and this is strand number 2. We have 2 strands. When there is adenine here, look at this, if you can see my hand. If this is adenine, it will fit with thymine in the other strand. And if this is cytosine, it will fit in with guanine. So I want you to remember it ATCG, because A go with the T, it fits in with the T, and C fits in with the G. How does it fit in? Like this. AT, like a wedge shaped, it fits with the T. C, let's say a spherical structure, it fits with the spherical structure of the G. And you have to remember this. A, T, C, G. This is how it fits with each other. So the nitrogenous bases are more inside. Uh, uh, you told us that beside the nutritional basis, we have deoxyribose sugar and phosphate. Yes, this is outside. The rest is going to be outside. It's arranged in this way specifically because you want the outside to contain the hydrophilic groups. So here is how it goes together. A, T, C, G. Look at this. A fits with the T. C fits with the G. How? Look at this. It looks like this. It fits with it. So if I give you a whole strand, you should know the corresponding strand. Like if I say A, T, C, G, what's it going to fit in with? So you will say A fits with the T. T fits with the A. C fits with the G. G fits with the C. So ATCG specifically. The inside is hydrophobic because of these nitrogenous bases, and the outside is hydrophilic, which is the sugar and phosphate, in order to keep these DNA in contact with the fluid of the nucleus. So DNA, genetic material, storing information about the two things that we talked about, inheritance and instructions for proteins. The DNA made of two, which is double, nucleic acid strands twisted together, double helix. Double helix is like this. Here is one, here is two, and they go like in a helix like this. They twist against each other like this. Here is one, and the other one is here. They twist with each other. What goes with the T? A, G, C. Chromatin is DNA with protein, and I mentioned that. During cell division, important, because chromosome is the same thing, but not during cell division. DNA is arranged in chromosomes. You have to remember this. How about chromatin? No, chromatin is an exception, only during cell division. So if I ask you, DNA is arranged into chromosomes. This is the normal arrangement. Chromosome change occasionally, become chromatin just during the cell division. What is considered segment of the DNA? It's the gene. The nucleotide sequence in DNA encode for the instructions. So remember I told you it's multiple nucleotides. The nucleotides are arranged in a specific way in order to tell you, or in order to contain the script of how to make proteins. I talked about this slide before, but you do need to remember it. We talked about it in nutrition before, when we talked about carbohydrates, fats, and proteins. But for proteins, here you need to know it. Here it is again. It's part of the structure and function. Regulate cell processes, enzymes, neurotransmitters, antibodies, hormones. All of these are proteins. Enzymes, hormones, antibodies, neurotransmitters, all of these are protein in nature, plus the structure and function of the cells. Now, if you are going to do protein synthesis, we already have the blueprint in the DNA, but the blueprint in the DNA contain a lot of genes. Instructions for what? Instructions to make all proteins. We don't do all proteins at the same time. We do one at a time. So I will take this piece here. Let's say we want to make protein number one. Give me instructions for protein number one. It's this part only. Okay, I'm going to take a copy of this and send it out of the nucleus to the factory of the protein, which is the ribosomes, if you still remember. We did that in the cells. So the manuscript, the original script, the blueprint, is the DNA, contain instructions for all proteins. You wanted to make this specific protein, get a copy of it, and the copy is called messenger RNA, and this copy can leave the nucleus and go out in the cytoplasm to go to the protein factory. What's a protein factory? Ribosomes. Yes, somebody have a question? Somebody have a question?
I think you have the hand icon up, Professor.
Okay, I don't know why. Oh, here it is. Okay. All right, thank you. So this is the first thing. So the question comes, if you have instructions for everything in the DNA, why don't you just send the DNA out of the nucleus to go to the ribosomes and give them the instructions by themselves? No, you cannot do that. Two reasons. Number one, DNA is the blueprint. It's the original script. It's too important, too expensive. You cannot send it out of the cells. It can be destroyed for whatever reason. You can't. This is too important. You cannot, if you are a manager in a factory, you're not going to send your blueprint out and expose it to different things. You're not supposed to do that. So instead, a specific that you're not going to do all the proteins at the same time. So all what you need to do is a copy. This is number one. Number two, even if you want to, DNA is too big to leave the nucleus. When you leave the nucleus, there are certain pores. These pores are too small, barely can allow messenger RNA, because messenger RNA is a copy of a little piece. So it can go through the pores. Those are the two reasons DNA itself does not leave. So messenger RNA, which is a copy of this piece that represents one protein only, is the one that's going to leave the nucleus, get out of the nucleus from the pores, and leave, go to the cytoplasm, reach the ribosomes, and start the process. So RNA, just like DNA, in DNA we said it's nucleotides. Here it is nucleotides, because both of them are nucleic acids. What's a nucleotide? Nucleotide is nitrogenous base, phosphate, and sugar. Same thing here, same structure. What's the difference? The difference is, remember, it's RNA because of this, ribose, ribonucleic acid. Ribo means ribose. It's just easier to say ribo instead of saying ribose nucleic acid. It's ribo, meaning ribose. So the sugar is ribose. How about the other one? It's DNA. So D is deoxyribose. This is one difference. The other difference is the nitrogenous bases. Remember, we have four that fits together, A, T, C, G. Here, instead of the T of the T, which is the thymine, it is actually uracil. RNA does not have does not know thymine. Instead, uracil. So instead of ATCG, it's AUCG. DNA, ATCG. Here, AUCG. The other thing is, remember I told you DNA, the D should remind you with double strand. RNA is a single strand. So here are the differences. DNA contain deoxyribose, while RNA contain ribose. The four bases are ATCG, here it's AUCG, and uracil is replacing thymine, and the configuration D is double, RNA is single strand. We have three types of RNA, and I talked about the first one, which is messenger RNA. You get a copy of this piece of the DNA that represents instructions for one protein. And because it's like a message that the DNA is sending out of the nucleus, going to the ribosomes, so we call it messenger RNA. This is the first one. The other two are going to be transfer and ribosomal. Ribosomal is the one in the ribosomes. This is where we make the protein. Transfer from the name. This is the one that's going to transfer the amino acids for you to make proteins. So three types. Messenger RNA is the first one, which is a copy of piece of the DNA. And we call this process of getting a copy of the piece of DNA, we call it transcription. Like you transcribe something, meaning you make a copy out of it. Ribosomal RNA, it's referring to the ribosomes. We talked about the ribosomes in the cells. And we said this is where we make the proteins. Yes, just a little bit more details. The ribosome, which is ribosomal RNA, is two units as you see here, small units and big units. And when this messenger RNA comes, it's going to go in between those two units. Transfer RNA from the name transfer. So it's going to transfer amino acids for you when you need it. So messenger RNA go here within the ribosome. Ribosome is the factory. Messenger RNA, this is the message containing instructions of how to make a protein. It will go put itself, the two units separate, and it will go between the two units, and you start reading it. And every three of those, it will represent one amino acid, each three of these units, like ACG, GTC, CG, it's not T, it's U, AUG, GUA, UGC, CTG, and so on. Every three represent amino acid. So it will fit itself in here, and ribosomes will call on the transfer RNA and tell the transfer RNA, bring the amino acids that those first three means. So the first step, if you wanted to make a protein, you need to make a copy from DNA to RNA, and we call that transcription. And we talked about these already. RNA is double strand, or single strand, I'm sorry. DNA is double strand, D goes with the D. The sugar in the RNA is ribose, while in the DNA is deoxyribose. Here it's ATCG, here is AUCG, ATCG in the DNA, and AUCG in RNA. And there are three forms of RNA. Messenger is the one that gets a copy of the genetic information of a certain protein from DNA. Leave the nucleus through the pores, go to the cytoplasm, find the ribosomes, and stick with it. Transfer RNA is the one that's going to transfer the amino acids. So the first process is called what? Transcription. When you transcribe something, you're making a copy out of it. And this is DNA. You make a copy of piece of the DNA, which is instructions for one protein. And we call this copy that will go as a message, messenger RNA. So you need to remember the name, transcription. Transcription. Then when the messenger RNA leave the nucleus, go to the ribosomes, you start to translate the code or the instructions, and transfer RNA start to get the amino acids for you. We call that translation. So you're going to translate. You're going to read the code on the RNA, and when you read it and understand it, you translate it, and then you see which amino acid fits with this, or what does this code is translated, or this code is translated into which amino acid. Translation into a specific amino acid. Remember, we talked in nutrition, we said proteins are amino acids. So you need to get amino acids, a lot of them, and we put them together to make a polypeptide, which will become protein. So here is the transcription again. When you transcribe something, means you make a copy out of it. And then you translate into protein. You make a copy of the instructions, and then, which is transcription, and then you translate into protein, or translate in a sequence of amino acid which make protein. So when you make a copy of this piece of the DNA and you see ATCG, remember, when you see the T, you cannot make it T. You're going to make U instead, because RNA does not understand thymine. It doesn't know thymine, doesn't have thymine, it doesn't have T. It has U instead. And then you're going to translate that. Where are you going to translate? Ribosomes, that's located in a cytoplasm. And what do you mean by translation? I translate the code on the messenger RNA into amino acids. So this is a transcription. There is an enzyme that's called RNA polymerase. What is this? RNA, this is a product. Polymerase is polymerization. This is the enzyme that's going to make RNA. Just remember it this way. That's good enough. And I'm not going to go too deep into uh, these processes. Here is what I want you to know. Remember the DNA. This is the DNA right here. It's two strands. Simply, you're going to separate the two strands. RNA polymerase is going to get a copy for you from a specific part of the DNA that represents one amino acid, which is the gene. What's a gene? It's the gene is a code or a transcript to make one protein only. DNA contain a lot of genes for a lot of proteins. What's the name of the enzyme? RNA polymerase. So simply what I want you to know, you unfold the two strands. Remember, the two strands are held together by the bases, nitrogenous bases, fitting together ATCG plus hydrogen bonds. So you're going to separate them, and RNA polymerase is going to help you to make a copy of certain piece that contain the desired instructions for the desired protein, which is a specific gene. So if you're reading the DNA and you find TGCA, what's going to make it in the RNA? When I see T, I will fit it with A. When I see G, remember ATCG or AUCG if you're doing with the RNA, if you're dealing with the RNA. So when I see T, I will fit it with A. I'll get the one that fits with it to make the RNA. Let me explain this part. When you do a copy of the DNA, you're not doing an exact copy. You're doing like a negative copy. What is this? Do you guys, I know we're in a digital age now and we don't use the old-fashioned camera, but if any one of you saw the old-fashioned camera when you take pictures and it give you a negative, they call it the negative, which is the opposite of what you actually have. Like your hair is black, in the negative it appear white. And if your body is white, it will give you like black or gray and so on. So the negative is the opposite. So same thing here. The copy here is not an exact copy. It's the opposite. So when you see T, you get an A. Why are you doing that? Why don't you just go straight forward and TGCA, just make it TGCU, and that's it? Because when you start the translation, you're going to do the opposing or the opposite of that. Just like you take picture, you have the negative, and when you print the negative, it will give you a copy just like the original one. So transcription is like the negative. The messenger RNA is a copy that looks like the negative. When you translate, when you print your picture, it will appear like the original one. So you're doing like an intermediary step. So when you see the T, it's A. When you see the G, C. CG, which is ATCG. A fits with the T. You have to remember this. A fits with the T. C fits with the G. ATCG. Say it several times and you have to remember it. ATCG or AC or AT, I mean AUCG. ATCG, AUCG. ATCG if you're making just a copy of the DNA into another DNA. But if you're making RNA, you have to go like the negative. So instead of ATCG, it will become AUCG. And you can be asked about this. So here I am making a copy here in the form of RNA. I see T. Okay, it's U. It's A, I mean. I see A. Can I make T? No, you cannot. It's RNA. It's U. About the C, normal. G, G, C, G, you will have C. You can be asked about this and you can get a sequence and ask you if this is right or wrong. So all what I want to remember here is ATCG. This is for the DNA. For RNA, instead of T, just put U.
Can I ask a question?
Yeah.
When the T turns into a U, that that information going from T to U, you say it's a copy, but it changes its letter. Is it an exact copy, or is the
No, and it's
T and
not
the
an exact
U
copy.
two completely different?
Yeah, it's not the exact copy. It's the opposite. Like when you get a picture and you get the negative.
Well,
So
I understand the T and the A are opposite, but
not
then
only that,
when
not
the
only that, you're
well,
making you're making the opposite of the whole strand.
yeah,
Like
but the part that stands out to me is what you're saying is the T and the U and the A. Those three, um, when they go into the RNA, the DNA goes into the RNA. When it goes from the T, or I'm sorry, from the A over to the RNA, it turns into a U. It's not a
yes,
T over there.
yes,
What is the difference between the T and the U when it gets over to the RNA? Why is it turning into a U?
because RNA doesn't have thymine. It doesn't know what's a thymine. It replaces it with its own version, something similar to it, in the RNA. Okay, this is part of the RNA. So they
So
found
it's
it out. There is nothing like a reason or something. It's just that the RNA doesn't know. It has its own version of T, which is U.
so it's not an exact copy going from the DNA to the
It,
RNA. That's not
it,
an exact copy.
it is. U is its version of. Let me give you an example here.
Oh, so that's when the translation comes in. It translates the T into a U.
No, no, not yet. Not yet. You're not
Oh,
translating. You're just getting a copy. Let me give you an example. Um, I don't know if you guys know that or not, but for like cars in Italy, there is a car called Fiat in Italy. Okay, Spain make the same car, but they call it Seat instead of Fiat. S is for Spain. What is the difference? This is called Seat and this is called Fiat. They look like the same, but this is the Spanish version of it. So U is a structure that's very similar to T, but they call it uracil. I don't know if you guys ever see that or not before.
well,
Lexus.
when you
Lexus
say
is Toyota. I don't know if you
when
guys
you
know that or not. Lexus is
when
Toyota.
when you say very similar, it actually isn't very similar. It's exactly the same. It's just named differently.
Kind of. Yes, I don't want to go deeper than that, but U is the version of T in ribosomal RNA. It is the same, but it has another name, so it's like another version of it. Okay? All right, so here it is. The brown is RNA, the blue is the DNA. So we're making a copy, which is the blue. I'm not going deeper again. I'm going to, if I see like more information that's more in depth, I'm not going to go more than what we need because it's going to be too much. So here it is. We get a copy, and who's helping us to make this copy? And making a copy is called transcription. Who's helping us? Which enzyme? RNA polymerase. Why it starts with RNA? Because we're producing RNA. So it's RNA polymerase, the enzyme that make a copy in the form of RNA polymerase. So remember this. So you get a copy right here, and this copy is going to, let me go back here. The copy is going to leave the nucleus, exit through the nuclear pores, and go into the ribosomes to be translated. But, and again, I'm not going to go in depth more than that. Before the messenger RNA leave the nucleus, you have to trim it, remove the unnecessary parts. Example, if I have DNA and this part contain like my hand here, this part of the DNA, let's say the whole DNA is like my whole arm. This part, like my hand, contain instructions for one protein. So this is a gene, an instruction for one protein. And this is what I'm going to take copy of. Here is a copy messenger RNA. The copy is not all full of instructions. There are certain pieces that are empty. I'm going to cut it off to make it even smaller so it leave through. So one more thing that I want you to know, after you make the messenger RNA, you need to cut the pieces that doesn't represent anything to make the size smaller. You just need the instructions. I don't need all of this. If there are no instructions here in the fingers, I'm going to cut it off. It's empty. So I will end up having this piece. It contains the same instructions, but without any extras. And here it is, transcription happened in the nucleus. Transcription is DNA to messenger RNA. What's the enzyme? RNA polymerase. Here it is. DNA, one of the strands is the one that we get a copy of. It's called complementary strand. This is the one that we get a copy of. So you unwind the DNA, means unwind it, the two, separate them, and you make a copy of the DNA in the form of messenger RNA. And then this copy, after you trim it to make it smaller, it leaves, go to the ribosomes. And in the ribosomes, you're going to do something called translation. And this translation consists of, you start initiation, get one amino acid, elongation, adding more, until the end, you're going to terminate. How are you going to do that? Here it is. The messenger RNA is in the ribosome now. Here it is. Messenger RNA is a copy of DNA. Now messenger RNA goes into the ribosomes. And now we are translating. I see ACU. This is triplet. Triplet. 3 bases. Each 3 bases code for 1 amino acid. So what does the ACU? You're not required to know the translation. Just understand that we translate. That's it. So this is a code. 1, 2, 3. Each 3 is called a code or codon. And this codon translation, it will be translated into a specific amino acid. These are just examples. You're not required to know it or memorize it. So I see ACU. It means threonine, amino acid threonine. Okay, move on. Next part. The next 3, CCU. It means proline. I will ask transfer RNA to bring the proline for me. Move. Go to the next part. Who is moving? Ribosomes are telling the messenger RNA, next three, next three, next three, or next codon, next codon. So one by one, code goes. We understand the code. Here is the first code or codon, ACU. Ribosomes are translating every three, triplet. That's called codon. So here is the first three. Let me read it. ACU. Let me translate. Threonine. Transfer RNA, bring threonine. Messenger RNA, move a little bit forward. Let me read the next triplet or the next codon. CCU. This is reading. Translation. Proline. Okay. Transfer RNA, I want you to bring proline. You bring the proline. And then each two, you will connect them with a bond, which is called peptide bond. It's covalent bond, but we call it peptide bond. And then ribosomes are going to ask messenger RNA, move forward to read the next. Here it is, GAG. It means glutamate. This is the translation. Transfer RNA, bring glutamate. Here it is. I'm going to attach it to the proline. And so the next is glutamate again. That's fine. Transfer RNA, who is responsible for glutamate? Bring it. Attach. Next, AAG, it means lysine. Transfer RNA responsible for lysine. Bring it. Attach. So now you're translating codon by codon by codon by codon. Translate it into amino acid, amino acid, amino acid, amino acid, and you're going to attach these amino acids to each other. So here it is. Each three is a block. Each three is a triplet. Each three is a codon. Okay? You don't need to know that. You can take a look at it if you're interested. So like UCU, UCU, that will be this one, which means serine. You're not responsible for that. I'm just telling you how it works. Now, when you start the translation, there is a codon that just means start, which is AUG. And there are three that means stop. So you see a big transcript, which is messenger RNA. It contains a lot of letters. Ribosomes need to know where should I start, and then I'm going to add one by one by one, translate, add, translate, add, translate, add, until I come to the end. How do I know that this is the end? There will be one of these codes that means that's it, you're done. Here it is. I'm not asking you to know these. Just AUG is the start, and there are three that means the end. And even that, just take a look at it to understand it. I just want you to know the concept itself. Now, I'm reading the first one. And the first one is AUG, let's say. This is messenger RNA, the purple. And here is that triplet or the codon. What does it mean? It means amino acid methionine. You don't have to know the names, just certain amino acids. I'm going to call the transfer RNA to bring it, which I will say, I am the ribosomes. I'm reading the messenger RNA and translating it. I'm reading AUG, translation, methionine. Okay, I'm going to call on transfer RNA. Who is the transfer RNA who's responsible for methionine? Me. Bring it. But I will double check to make sure that this is actually fits. So there is something called anticodon that's on the transfer RNA. So it will fit in together to make sure this is the correct one. Which transfer RNA is responsible for methionine? Me. Okay, come over here and bring methionine. Should I leave methionine and go? No. I want you to fit yourself first with your anticodon. So your anticodon of the transfer RNA, I want you to fit it with the codon of the messenger RNA to double check before you leave. Did you double check? Yes. Is it the correct one? Yes. Leave methionine and go. And here it is. This is the ribosome. Inside, the green here is a messenger RNA. So you started with this one. So next one, AAA. This is a messenger RNA. Ribosomes is reading. It means lysine. Okay, here is the yellow transfer. Transfer RNA. Which transfer RNA is responsible for lyse or lysine? Me. Should I leave lysine and go? No. Fit yourself in first. This is AAA, U. Yes, that's correct. Leave lysine and go. And then I go to the next. GAU. Ribosomes is reading. Triplet. Every 3. This triplet means aspirate, for aspirate. Okay, which transfer RNA is responsible for that? Me. Should I leave it? No. Fit yourself with your anticodon to the codon to make sure that you are bringing the correct one and leave it. And now look, one after one after one, these circles, pink circles are amino acids. When you're done with one and you bring the next, you have to connect them together. How to connect them? It's a covalent bond that's called peptide bond. Covalent bond called peptide bond. So amino acids, covalent or peptide bond, another amino acid, covalent or peptide bond, another amino acid, and so on. It goes all the way in a chain like this. So because of this one called peptide, so when you see this sequence here, we call it polypeptide. What does it mean? Amino acid, peptide bond. Amino acid, peptide bond, which is covalent. Amino acid, peptide. So do you have several peptide bonds connecting the amino acids? Yes. Then we call that polypeptide. What's a polypeptide? A long chain of amino acids connected by peptide bonds. So here it is one more time. Start initiation. You will see the code that means start. And then the first code will be translated. Transfer RNA, bringing it. Fit yourself in. Leave it. Next. Here it is. Okay, fit yourself in your anticodon of the transfer RNA with the codon of the messenger RNA. Fit yourself. Did you fit yourself? Yes. Leave it and go. And then I'll connect them together. And next. Initiation followed by elongation. Elongation is adding amino acid after amino acid after amino acid and connecting each 2 successive amino acids to each other. And we call that elongation, making a polypeptide. Until you see the stop codon and you're going to terminate the process, you're done making the polypeptide. So translation, where does it happen in a cytoplasm? Where in a cytoplasm? Ribosome. There are 2 units of the ribosome. This is where we make the proteins. The 3 letters are called codon in the messenger RNA. What's a codon? A codon is a triplet of bases that means 1 amino acid. Transfer RNA that's transferring or bringing the amino acid. It has an anticodon. So if you see a question, what's the difference between codon and anticodon? Codon is the triplet in the messenger RNA. Anticodon is the triplet in the transfer RNA that fits with the codon of the messenger RNA. And then amino acid attached by peptide bond. Because it's a lot of peptide bond, we call it polypeptide. Polypeptide will become protein at the end. We talked about this before for the proteins. And these are the different functions: enzymes, hormones, neurotransmitters, antibodies, structure, and so on. Okay. So next is reproduction. And reproduction, we're not going to talk much about that. We will discuss it later. But reproduction here is when you reproduce, like one cell will become two. You're making a new generation. And it is two steps or two cell divisions. One of them is called mitosis. We have a lot of details to talk about later, but this is an introduction to this part. So mitosis, it's coming from the Greek mitos, and mitos means thread. Why did you call it thread or mitos? Because this is one cell. I'm going to double the contents, and then it's going to divide into two new cells connected by thread. And threads are going to attach to the two daughter cells, and they will be pulled apart. So this mitosis happen in the somatic cells, not in the germ cells. What does it mean? All cells in your body are called somatic cells. The only one cell that's not called that name, which is called the germ cells, these are the sex cells. Sperm in males, eggs or ova in females. So what's my skin cells? Somatic cells. Liver cells, somatic cells. Brain, hair, digestive system, reproductive system, urinary system. Every cell in your body is somatic, and somatic means body. Soma is body, translation. So somatic cells are all your cells are going to use this one to reproduce, mitosis. And it happens with most of our tissues. Meiosis, on the other hand, meiosis is coming from Greek myos, and myos means uh, reduction, less, reduction. Why? Because in mitosis, we have 46 chromosomes, 46, which is 23 pairs. And this 46, which is 23 pairs, when it divide into two, it duplicate first and then divide. So the daughter cells will have the same number as the parent cell. They just attach by threads, and that's why we call it mitosis. This is completely different from your germ cells or sex cells like sperm and ova. How is it different? Because you start with 46, and then when you divide, you end up having a sperm that contain 23 only, not 23 pairs, not 46, 23. Why are you reduction like this? Why the sperm contain 23, which is half? Why the ova have 23, which is half? Why don't you have 46 like what you did in mitosis? No, it has to be like this because later on, the sperm, which contain 23, will merge with the ova, which also contain 23. So both of them together become normal cell, which is 46. So this is the main difference between mitosis and meiosis. There are other differences. This part is not easy. I understand that. That's why I'm recording. But we will discuss even more details. For now, mitosis, you start with 23 pairs, which is 46. You end up having two cells, each one contain also, just like the parent, 23 pairs, which is 46. And this happen in all cells. Because you lose skin cell, you want to make another skin cell, which is identical. But for meiosis, this is for reproduction. This is completely different. You wanna make offspring. You cannot make, you start with 23 pairs and you have 23 pairs, you can't do that. You start with 23 pairs, you end up having 23 singles in the sperm and 23 singles in the ova. When they come together, they will become 46. We will talk about that more. But here I said I'm going to duplicate. So somatic cells, this one, mitosis. I start with one cell. Let's say this is a skin cell. How many chromosomes do you have? All of them contain the same number, 23 pairs, which is 46 chromosomes. So what do you want to do? I want this cell to produce two. Each two should contain the same number. So you have to duplicate first. So instead of having 23 pairs, I will end up having two 23 pairs. So when it divides, the daughter cells looks exactly like the original cells because this is not reproduction. It's not germ cells. Skin cell, I want to make two skin cells identical. So you have to duplicate first. And in order to duplicate, you duplicate everything, including the DNA. DNA duplication is called replication. Remember, the cells contain nucleus and contain organelles. Inside the nucleus, you have DNA. So you have to duplicate everything so that you end up having two daughter cells identical to the original cells. And in order to do that process, you have to duplicate the DNA itself. You have 23 pairs, which is 46. You wanted to make it two sets of those, 23 pairs and another 23 pairs. Each one will go to one of the daughter cells. You call this process of duplicating the DNA replication. You have DNA and you copy it into DNA. This is completely different than what we finished, which is a transcription. DNA, you make RNA out of it, and it's part of it. But here, it's the whole entire DNA, and you make a whole copy entire DNA. Completely different. But you start the same way. You unfold first, you unzip or unfold the parent DNA. And then instead of making a copy of part of the complementary strand, no, you make a complete copy of one strand and a complete copy of the other strand. You end up having two sets, just the same as the parent one. Here is how we do it. You start with one, separate them, make a copy of the original strand here, make a copy of the complementary strand here, and then attach to each other. So you started with one, you ended up having two. This is how it works. Why aren't you like separating them and make two copies of this one only? Because this process is called semi-conservative. Semi-conservative. So you end up having one blue and one yellow. So you got half. And then the other one is one blue, one yellow. So it's semi-conservative. Why are you doing that? Because the two copies are similar to each other, but you wanted one of the original with a copy and one with the original of the copy just in case if something happened. So here it is. Those are the two copies. And remember, genetic code is written here. And they go opposite of each other, like this. Like this are the two strands, they go like this. They call it the 5 to 3 and 3 to 5. You don't need to know more details about this. This is good enough. But the enzymes that help you to do that here is something that you need to know. Enzymes. Remember, the only enzyme that we talked about in the RNA is RNA polymerase. Remember, RNA polymerase is an enzyme that make RNA. That's why it's called RNA polymerase. The other enzyme that here that make a copy of DNA, what's a product? DNA. So we call it DNA polymerase. This is one enzyme. The enzyme that unzip the double helix, the two strands, unzip it, helix, the two helix separate, they call it DNA helicase. The enzyme that help us start is called primase. The one that remove parts of the RNA is called exonuclease. And the one that ligates or fix them together is called ligase. So look at this. You start with helicase, separating them, and then primase to give you the prime to start. And you move in the opposite direction like this. DNA polymerase is giving the copies for you. And then you end up having pieces, and these pieces are going to held together by something called ligase. See, so cellular reproduction, you make babies. Mitosis and meiosis are different. Mitosis is for somatic cells. Meiosis is for germ cells. So in somatic cells, you do mitosis. In germ cells, you do meiosis. Cell division start with DNA or RNA. Of course, it's DNA. RNA is in protein synthesis. DNA, is that conservative or semi-conservative? It is semi-conservative. What does it mean? Right here. It's semi-conservative. One original with copy and another original with copy. You're doing that on purpose. You don't want to put the two blue together and the two yellow together just in case if something happened. Because if something happened wrong in the yellow, you have the original one here and it can fix it. And that's why it's called semi-conservative. You need to know that, semi-conservative. Which enzyme unzips? Helicase. And which one is simple? It is polymerase. Now, a little bit about the proteins. We need to do that. Proteins. Remember, we talked about the proteins before, and we said the protein is a polypeptide chain of amino acids, and it has a lot of functions. And we said like enzymes and hormones and uh, uh, structure, like here, like collagen, transport, and different functions. And I mentioned already that you get one amino acid and you put peptide bond, which is type of covalent bond, with another amino acid. You call that dipeptide. And you add, you call it polypeptide. Now, this part is important, including the pictures. So understand this part. It is not that simple that you make protein. What I told you already, remember when we make DNA, we did transcription, messenger RNA, leave the nucleus through the pores, go to the cytoplasm, find the ribosomes. Ribosomes are going to translate each codon, which is a triplet, into amino acid, and then you attach the amino acid by peptide bond, which is a covalent bond, and then attach one by one. This is two dipeptide, and then add more. You call that polypeptide. Are you done? No, I'm not done. It's not just simple like this. Making protein is like making a house. How? In your house, if you are a builder and you're going to build, you get brick, you put cement, another brick, cement, brick, cement, brick, cement, brick. You end up having just a wall, one wall. Is that a house? It's not a house. It's just one wall. Then you make another wall and another wall. Now you're adding up until you end up having a whole room. Let's say I finish the bedroom, and then you make another bedroom, and then you make another a bathroom, and then you do a kitchen, and so on. And you end up having a whole entire house. Same way we do with the proteins. It's primary structure, secondary structure, tertiary, and quaternary structure. This is the one that we talked about, the first one. Just a polypeptide chain, amino acid, peptide bond, amino acid, peptide bond, amino acid peptide bond. This long chain is called the polypeptide. Not enough. You start to fold it like this. Make it either a pleated sheet like this. Look, bend, 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 or alpha helix. This is called the secondary structure. And then you fold it more like as if this is like a wall, and you're bending the wall to make another wall beside it. And when you have all walls and the ceiling and everything, this is one room, which is a tertiary structure. Now, when you put all these rooms together to make a house, this is quaternary structure. So look at the primary structure. Primary structure, you get one amino acid, peptide bond, which is covalent, another amino acid, peptide bond, another amino acid, until you get this chain, a long chain of amino acids. We call that primary structure. And then the secondary structure is this long chain. You fold it and you put it in the form of either the top one here, which is the alpha helix, or pleated. One of those. You have to put it in one of those forms. This is called the secondary form. How are you keeping it in that shape? Why it doesn't unfold and straighten again to go to the primary? Hydrogen bonds. Hydrogen bonds are keeping it in this secondary structure. And it can be alpha helix or pleated sheet. Then you put it in the tertiary structure. Three-dimensional structure now. So again, like you're building a house, you put bricks beside each other, you make a wall, you bend it. Not a room yet. This is secondary. And then you make another two and you put a ceiling and a floor. Now you have a whole building. This is a whole room, I'm sorry. This is like tertiary. And then multiple rooms, this is quaternary. So here it is again. Polypeptide chain, just a chain, a long chain of amino acid in a polypeptide form, this is primary. When you fold it in a form of helix or pleated sheet, this is secondary, like you're making, bending the wall. And then when you finish making a whole three-dimensional structure, this is a tertiary form, and they call it, this is a functional form, like a whole room. It do the function. The room do the function. Like this is a bedroom, it do the function of you sleep in this room, and so on. Quaternary, here is one room, bedroom, bathroom, kitchen, another bedroom. When you have all four or multiple rooms together, this is called the quaternary structure, and this is the final product. You don't make a wall and tell me this is a house. It's not a house. So, and you don't get a polypeptide chain and you tell me this is a protein. It's not a protein. This is just a polypeptide chain, which is just primary structure. You have to make it secondary, tertiary, quaternary. And the best example for quaternary structure, which is a whole protein, is like hemoglobin. Hemoglobin is heme, which contain iron, and globin is the protein. Look at the protein, like one room, two room, three rooms, four rooms. Okay, you put these tertiary, 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 room, 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 room. You put them together in order to make a whole house, which is a quaternary structure. If you expose the protein to heat, it become denatured. What does it mean? Here is what I mean. If you get an egg, I think you guys know that the white inside the egg is protein. So if you open a raw egg, just an egg, you will see like semi-fluid, semi-liquid. Like if you open it, it will fall, right? It's not solid. But what happened to it when you boil it or when you expose it to heat? The nature change. It lose its nature. It becomes solid, right? We eat it. Don't you eat the boiled egg? Do you eat something solid or liquid? It's solid. So what happened to the nature, the original nature? When it was an egg, without exposing it to heat, it was semi-liquid. What happened to the nature? You changed the nature. We call it denatured. And once it's denatured, it will never return back to the natural form. Or show me, like get an egg, boil it, become solid. Can you change it back to the semi-liquid? Of course not. You cannot. Once it is denatured, it will never go back to the natural form. Temperature do that, and I can tell you if I get the same egg and add some acid to it, like if I get some acid from the battery and put it on the egg, it will do the same change. It's not just heat. It's also pH, changing the pH, which is like acidic or something. So here it is. This picture is very important. We can ask you about it. So you need to understand, this is primary structure. Those two purple here, secondary structure. This one here is tertiary, and the final form is quaternary. Remember, this picture is important, and we can ask you about it because it's in the core of the chapter. And here it is. Protein is made of what? Amino acids. DNA is the one that's determine the triplets that it contains. It's in a specific sequence, and this sequence determines the sequence of amino acids. So the sequence of the amino acid is primary. When you fold it into either helix or pleated sheet, you call it secondary. When you coil it and loop it more and folding it into a three-dimensional structure, we call that tertiary. And when you have multiple of these tertiaries, multiple polypeptide in the form of tertiaries, we call it quaternary. And if you expose protein to heat or pH, we call it denatured. Here it is again, primary, secondary, tertiary, quaternary. And this is very important to know. And that's it. The rest is a summary of what we talked about. So I'm going to stop here. Stop recording.